Pariser. I'm the CEO and founder of Humans for Education. And this is Stephanie, our program director of Humans for Education. And this is our third episode. And today we are your hosts.
So today we're very excited to be joined by Dr. Stephen Dewhurst. Dr. Dewhurst is Vice Dean for Research at the School of Medicine and Dentistry, Associate Vice President for Health Sciences Research, and is Dean's Professor and Chair of Microbiology and Immunology at the University of Rochester. Dr. Dewhurst has over 25 years' experience as a molecular virologist, working on both DNA and RNA viruses, including HIV and influenza. And now he can add coronavirus to that list. And during this podcast, you'll hear what researchers like him are doing to help answer some important questions about the virus. So thank you so much for being here today, Dr. Dewhurst, and we really appreciate you taking the time to speak with us and answer some questions for our listeners.
Well, thanks very much for having me. It's really a great opportunity, so I very much appreciate it.
We're very excited. But before we get into the questions about what's going on at the University of Rochester, including COVID, we want to talk really quickly about why we're doing this podcast. So Humans for Education is doing this podcast to basically feed families in Kenya because of COVID right now. So if you like our podcast and if you've been listening, please feel free to support some of those families by going to humansforeducation.org/COVID19. And you can also buy masks if you don't have them already through our partner Mansa Wear, and you can find them on our website as well. So, Stephen, we're very excited to talk to you, and I'd love if you could tell us about some of the research that's going on at the University of Rochester right now that's related to COVID.
There's quite a lot of different things going on. So some of what's going on has to do with developing new tests, both for the virus, meaning are people actually actively infected, and also for antibodies, which obviously we hope is an indication of possible immunity, although we don't know that yet. So some of the virologic tests are, for example, in biomed engineering, Jim McGrath has this silicon pore system that is able to precisely capture viral particles like a sieve, really. And they're trying to develop that as a test for virus presence in biological fluids like blood or potentially saliva, which is emerging as a really important diagnostic substrate. So that we started out with nasal swabs, and those are, I think, people who've experienced having a nasal swab, they're not much fun. And they're actually quite hard to do right as well. So you can get a negative test result because you didn't sort of stick the little thing up all the way up somebody's nose. So it's sort of intrusive and more technically difficult than you'd think. So saliva, in any case, is emerging really as an important substrate. We have people in our Eastman Institute for Oral Health who have an interest in obviously dentistry, but if you think about that for even a second, right, you're in somebody's mouth where saliva contains virus, where you certainly can't mask somebody undergoing a dental procedure, but nor can you stop providing that service, right? Lots of people have emergency dental needs. Those have not stopped in the entire period of let's say, the shutdown we've had in New York State. So how do you keep people safe as patients, and how do you keep providers safe, dental care technicians and so on? And part of that is being able to measure infectious virus in saliva. So we're working with them to try to develop a sensitive assay for infectious virus. That's a little different than many of the other RT-PCR-based tests for viral RNA, which are agnostic with respect to infectivity. We've got other people like Ben Miller, for example. He has a company called Adaza. Adaza uses this arrayed reflectance technology, and I think you've already spoken with Ben about that, so I probably won't belabor that. But if people want to go back to a previous podcast and learn about that, it's a really exciting technology that is very different than traditional ELISAs, much faster, and potentially a lot more sensitive and able to look at multiple things at the same time. So those are some examples. We have other tests. Martin Zand, for example, is developing a finger stick test. So if you want to think about how do you bring research into the community directly, how do you have people be able to get a test without ever coming into the hospital, finger sticks are perfect, right? It's a tiny volume of blood. You can put it on filter paper. And if you have a very sensitive assay, you can use that to evaluate the presence of antiviral antibodies. And as we move forward, those kinds of tests are going to also be increasingly important.
And sorry.
No, go ahead.
For our listeners who are not super familiar between the difference with having a test that's testing for the virus and a test that's testing for an antibody, can you kind of explain the difference of why each of those tests are important to be developing kind of alongside each other?
Yeah. So antibody tests are kind of like in the rearview mirror. So they tell you, have you ever been infected with the SARS coronavirus 2? And we hope that if people test positive for antibodies, then they might be protected in the future, though actually we don't know that. So that's going to be important over time in looking at workforce issues and looking at people who may be susceptible and at risk. And it's important in knowing, right now, most important in knowing what is the community spread of the SARS coronavirus. So where we live in upstate New York, it's about 3% of the population have been infected. Down in New York City, it's more on the order of 20%. Those numbers get to be really important if you start to think about what's the possibility of a second wave of coronavirus infections, what's the overall vulnerability of a community as a whole. The tests for viral shedding are a little different, right? So those are much more relevant if somebody has symptoms and they, maybe they have fever, maybe they have a cough, maybe they have a loss of the sense of smell. Those are some of the features of coronavirus infection. There are others too. But you'd really like to know, then, are those folks infected with coronavirus, or do they have something else? And in many cases, people have something else. But it's really important to be able to rule that out, and also to know, does somebody need to self-quarantine? Do they need to stay home? And so that test is really important for anybody who's been exposed to get a quick answer. Are you potentially infected, and should you be self-quarantining? And what do you do in terms of protecting the people around you? Because what you don't want to have is people who are shedders of the virus showing up in workplaces or in public spaces, potentially transmitting the virus.
Right. So in addition to some of the research initiatives, what programs or infrastructure are in place currently at the University of Rochester that are helping support researchers answer some of these questions?
Well, one big thing that we've repurposed is we have a biological safety level 3 laboratory, which traditionally we've used for tuberculosis research. And TB is another respiratory pathogen. So it's not super difficult to repurpose that facility to really focus on the SARS coronavirus. So we've obtained isolates of the virus that we're using to, for example, test novel antivirals. So there's several groups at the university pursuing antiviral strategies. And one strategy that several folks are looking at is to repurpose existing drugs. So drugs that we know are already safe, they've been used in human trials or actually for full-on human use for other indications. Those are really attractive if you can repurpose them, if they have antiviral activity. And the groups at U of R that are pursuing that have some slightly different approaches and mechanisms that they're exploring that maybe will allow us to look at candidates that haven't already been considered by other people. So one of the approaches, for example, Josh Munger is a cytomegalovirus researcher, and he's really interested in host cell metabolism and how does the virus hijack host cell metabolism. But if you take the same principle, you can apply it to lots of other viruses. So it's true for flu and it's true for SARS coronavirus. It needs the host cell to reproduce itself, and it alters host cell metabolism to do that. And if you can interfere with host cell metabolism, what you have potentially is an antiviral that you'll never get resistance to, that being one of the concerns with other antiviral drugs. So that's an interesting approach that Josh and several others are pursuing, for example. We're also doing, very involved in clinical trials. There are multiple clinical trial networks at the university. So one is the Vaccine Treatment and Evaluation Unit that's headed by Angela Branch and Ann Falsey. And that's really been at the forefront and will continue to be at the forefront of COVID-19 trials. So the Remdesivir trial, the trial that led to FDA authorization of, to date, the only treatment we have that is known to be effective for COVID-19, that trial was conducted at U of R and many other sites nationally as well, but we're really part of that. And in the same way, that same group is also part of the vaccine trials. So Anne just opened the first vaccine trial in Rochester last week, and that's really one of the very first national trials. We have another bunch of trials that are teed up behind that in terms of vaccines, one of which will be to take a candidate developed at the University of Oxford. It's an adenoviral vectored vaccine. That one I think is really quite exciting, and we will be the lead North American site for that trial. So that's I think something to be really interested in. That one works very well in preclinical model systems, so it looks very promising. We'll find out. So that there are many ways in which we're involved, and so some of the infrastructure then is these large network awards that are really oriented towards testing new therapies and treatments. And one of the things that we have to look for, sort of in forward looking, is how do we expand our clinical trial capacity? I don't think we're going to lack for volunteers. People are going to, I think, be very interested in participating in these vaccine trials. So the bottleneck isn't going to be that. It's going to be, do we have enough people who can actually conduct the studies? So clinical trial coordinators, it's a very specialized skill set. And we're looking at, can we have a workforce development program to rapidly bring on board more people who can do that? Also figuring out where it's possible, how much can we do where we conduct the trial at a distance? So we don't bring people in necessarily to the medical center to do every single thing we need to do. Are there ways that we can do more of this at a distance? That also expands our ability to be able to run multiple clinical trials. In addition, we're involving several different networks. So the VTEU network, the one that Anne and Angela are involved with, is one network, but there's also a HIV clinical trials network that's international that Mike Kiefer is the principal investigator of here. That whole trial network's being repurposed for COVID. So they'll be taking on studies. They'll be doing actually a study with hydroxychloroquine with a second drug. So it's actually a 2-drug trial. And that trial will be initiating within the next month. So that's another way that we can really do more and be more involved in running trials.
Is one of the biggest reasons that you're having issues in terms of bringing more scientists on board in terms of the clinical trials, is that because of social distancing, or are there other issues with bringing more people on board?
It's actually just workforce, right? It's a specialized skill set. Let's say the clinical trial coordinator, and we don't have, and it's a national shortage. It's not Rochester. So training more folks who can do that and doing it quickly is part of the solution. And then, we're very fortunate here that we have the Center for Health and Technology that Ray Dorsey runs with Erica Augustine. And that center is really very interested in redesigning clinical trials, rethinking clinical trials. And that's part of what actually this crisis has precipitated, is being innovative and willing to really think of very different approaches. So in the same way that we've taken classes online, how much can you take consenting online? How much can you take running clinical trials remotely so that you bring the trial to people's homes and communities, and you don't expect them to come into the medical center, at least certainly not to the extent we have in the past. And those are actually really big opportunities that will last long after COVID is done. That transformation of the way we do trials, that's here to stay, I think.
So can you tell us a little bit more about Remdesivir and kind of how that works and what's going on with that?
I can tell you a little bit. So I should say that I'm not the world's most expert in terms of RNA-dependent RNA polymerases, although we have done some work with those enzymes and similar enzymes in the past. So one of the interesting things about coronavirus is it's a really huge RNA virus, right? It's a 30 kilobase genome. And most RNA viruses are just nowhere near that big. And one reason is that RNA viruses use polymerases to copy their genome that are error prone. And after a certain size, they make too many mistakes and the genome just becomes damaged too easily. It becomes mutated. Coronavirus has an editing function, so it has an exonuclease activity in addition to a polymerase activity. And that means that that's the reason that it's able to synthesize the 30 kb genome. It's also the reason that coronavirus is not as rapidly mutable as, let's say, influenza or HIV, right? Those are viruses, when we think of them, they have really high mutation rates, and they evolve very rapidly in response to host cell immunity. I would say one of the fortunate things, if there is any fortunate thing, is that the coronavirus is not as able to generate that level of mutation because its polymerase has this proofreading activity. So that's a little bit of a blessing. It's also a complication in developing antiviral approaches. So remdesivir was not produced to specifically target the coronavirus RNA polymerase. It just turns out to have activity against that in vitro assays. And one of the reasons that targeting that polymerase is a little complicated is this proofreading activity. So unlike, let's say, HIV, where if it was to misincorporate something, it has no way of removing that mistake. It just misincorporates and chain termination, as it's called, happens. So the chain is just broken. Coronaviruses can remove potential mistakes as they're synthesizing their RNA. So in other words, if you throw in there in essence, a fake building block of RNA, which is sort of what remdesivir is, it has a way of chewing that out. In most cases, remdesivir is chemically a little unusual, and it turns out it has a hard time removing remdesivir after it's incorporated. So I think there's a lot of interest, obviously, in getting more specific drugs. The appeal of remdesivir is it's already known to be safe in human beings, right? And so you've shaved a lot of time off in terms of developing a therapy. And that I think is going to be one of the limitations, right? That we may well end up with much better drugs, but if we have to do all the traditional safety testing in human subjects from scratch, that's much more challenging than repurposing a drug that we already have that maybe isn't perfect, but would be something that we can throw at the virus right now. Same with vaccines, that there may be more perfect vaccines that we will develop over time, but right now we need something that we can validate quickly and scale up and distribute very quickly, right? And we're looking at billions and billions of doses, so it's really not trivial about the scale.
Yeah, and if our listeners are interested in hearing more about clinical trials and vaccines and drug trials going on at the University of Rochester, we are speaking to Ann Falsey and Angela Branch in a few weeks, so look out for that podcast. Steve, data shows that the communities of color and low-income communities are really disproportionately affected by COVID-19, both in terms of morbidity and mortality. So can you tell us some of the ongoing programs that are going on in these diverse communities that are most affected by COVID-19 in terms of reaching out to these communities for research and for outreach?
Yeah, so one of the first things is really to remember that for research to be effective that involves community members, it's community participatory research, and that's really the important word, participatory. So it is a mutual benefit that's going on. Like, researchers have important scientific questions that are aimed at public health, and communities have their own needs, and understanding what the needs of those communities are and having trust. Right? Probably trust is the most important thing. So our Center for Community Health, that's run by Nana Bennett, it is unusual for the University of Rochester because it's located right downtown in an urban setting. It does a lot of outreach directly into the community. And that's necessary to build the trust that when you have a vaccine, like a COVID-19 vaccine, that we're able to test that in all populations. It's not much use if the vaccine turns out to work great in white men, but we don't know that it works well in communities of color, or that we're unable to establish the trust necessary, because there's the second part of this. The first part is testing the vaccine, making sure it works in everybody. And then there's the second part of when we get a vaccine, and that's a when, not an if. How do we get that out to every community? How do we make sure that all communities, groups, and individuals are ready and able to receive the vaccine? And that means trust also. I'm not speaking about the anti-vaxxers that's a whole separate conversation. I'm talking here about communities that that need access to this vaccine for their health. We need to have their trust, and we need to have programs that effectively get the vaccine to people who clearly are not getting adequate health care. And again, that's a whole separate discussion of the disparities that exist in the US healthcare system. That the COVID has really revealed some profound fault lines in our society and in the way that we allocate healthcare. But to be specific, Nana's group has a history of working with diverse communities in Monroe County, which is the county surrounding Rochester. And historically, the rates of immunization for influenza and pneumococcal disease have been very high in this community, among the highest in New York State, among the highest in the nation. And that's a function of a very deliberate, intentional outreach to provider communities to reach out then into the communities in which those providers are located, and specifically work with communities of color and diverse communities, so that the rates of vaccination in those groups have been very high also. That's the kind of thing we're going to have in place, both locally, but also obviously nationally. Again, it won't do as much good if we have an effective COVID vaccine, but not everybody gets the vaccine. What we can't afford to have is pockets of vulnerability. We need everybody to have access so that we have complete herd immunity across the entire society in every single community.
Super important.
Yeah.
So I know we're at a little bit over time. So is there anything else that you want to add in terms of gaps of research or knowledge or anything else that you think is important for our listeners to know?
Hard to say. I mean, I'll maybe slightly diverge from the question you asked directly, and maybe to say that this is COVID-19 is going to be with us for quite some time. I think staying the course is one of the most important things, right? So social distancing will be with us for a long time. It's not just a matter of personal health. It's a matter of public health. It's a matter of health of loved ones. I think that another really important thing is that there are many people who are at very high risk for COVID. There are others who perhaps are at lower risk. There are many people for whom this is really provoking a lot of mental health challenges. I think this whole social distancing, isolation, fear of contagion. And so my point in saying any of that is to say that kindness is actually probably the most important thing we can do right now. We are waiting for a vaccine. We will have one. But until then, kindness and thoughtfulness of each other is the most important thing.
Super well said. And I really appreciate that, because I think that is, while we're all sitting here and waiting for you guys to do such amazing work, with the research, I think that's the best that we can all do, just to each other. Yeah. So thank you all for listening to our Let's Talk Science podcast. Thank you, Stephen, for being here and providing some seriously awesome information. And if anybody is interested in helping feed some of our families out in Kenya who are definitely struggling with serious COVID issues right now, you can go to humansforeducation.org/COVID-19, where you can also find information on how to get yourself masks. And so please, if you have any questions, you can put them in our question box. And we look forward to seeing you next week. So thank you, Steve, for joining us.
Thank you so much.